Welcome back, everyone, to another deep dive. This time we're focusing on those fascinating little gatekeepers in our cells, voltage-gated calcium channels, or Cat2 plus channels for short. We're going to try to uncover really just how they take those electrical signals and kind of translate them into actions in our cells.
Yeah, and you've got a lot you want to cover. You're interested in the different types of channels, what makes them open and close, what roles they play in, things like how our muscles contract or how neurons talk to each other, and even how different medications can target them.
It's like we're on a mission to follow that calcium trail and see how much these channels really impact what our cells do.
And that's super important because the calcium ions are involved in so many critical processes in our bodies. They're basically like tiny messengers, helping to make things happen.
Okay, so before we get too far into all that, can you back up a bit and explain what exactly voltage-gated calcium channels are?
So there are these proteins embedded in the cell membrane.
Which is like that barrier between the inside and outside of the cell, right?
Right, exactly. And you can think of these channels as like guarded doorways.
Okay, so they're controlling what gets in and out.
Exactly. In this case, they're specifically controlling the flow of calcium ions, so they stay closed until they get, well, a specific signal.
A specific electrical signal.
You got it. So when that electrical potential across the cell membrane changes, it's like flipping a switch, causing the channels to open.
And then the calcium can rush into the cell.
Exactly. And it's all about, the voltage, hence the name voltage-gated, right? Now, there isn't just one type of voltage-gated calcium channel. We've got, a bunch of subtypes.
Each with its own, personality.
Yeah.
Characteristics.
Pretty much, yeah.
Yeah.
Different cells need different types of channels, right? It depends on their job.
That makes sense.
So, for example, we have L-type channels.
Which are involved in muscle contraction.
Yeah. And they stay open for a while, and they need those high voltage changes to trigger them.
Gotcha.
Then there are N-type channels.
Which are more involved in communication between neurons.
Exactly, yeah, because they help with, releasing those neurotransmitters.
Right, the chemical messengers that allow neurons to talk to each other.
Exactly. And just like L-type, N-types are also activated by those high voltage changes.
Interesting. So what about PQ-type channels? I know they're also involved in neurotransmitter release.
Yeah, you're right. They're pretty similar to N-type channels in that they're triggered by high voltage.
So it seems like we have multiple channels doing similar jobs. Is there a reason for that?
Yeah. It's like a backup system, just in case one fails. But also, they have subtle differences in, their properties. It allows for more fine-tuned control, Then we have R-type channels that kind of regulate how excitable neurons are. And finally, there are the T-type channels. These guys are a little different because they're activated by lower voltage changes and they don't stay open as long. They're involved in things like our heartbeat and how often neurons fire.
So it's like a whole relay team, right?
Yeah. Exactly. A relay team of channels, each with its own special role to play. And that diversity is super important because it allows cells to respond to electrical signals in very specific ways. Now, you are particularly interested in how that calcium influx, the movement of calcium into the cell, drives certain processes.
Right, like how it all works with neurotransmitter release and muscle contraction.
Okay, so let's start with the neurotransmitter release. When that nerve impulse reaches the end of a neuron, it triggers the N-type and PQ-type channels to open, remember?
Mm-hmm.
Well, that influx of calcium is like a signal telling the neuron, hey, release those neurotransmitters.
And those go into the synapse, the tiny space between neurons.
Exactly, and that's how they, communicate, allowing us to think and feel and move.
So without calcium channels, our brains would be in trouble.
Oh, big time, yeah. Now, let's talk about muscle contraction. That's where the L-type channels come in.
The ones specific to muscle cells.
Exactly. So when a muscle gets the signal to contract, it triggers those L-type channels to open, and all that calcium basically sets off a chain reaction that makes the muscle fibers shorten, so they generate force.
Okay, I think I'm following.
And this whole process is super dependent on calcium doing its thing.
Gotcha.
And it's not just about what type of channel it is, but also where they are in the cell. Like you'll find those N-type channels hanging out at nerve terminals, ready to release neurotransmitters, while the L-type channels are in those muscle cells, ready to make them contract.
It's like everything has its place, even at the microscopic level.
It's amazing, right? It makes sure the calcium goes where it needs to go and triggers the right response at the right time.
So, what happens when these channels don't work the way they should?
That's a great question, and one we should definitely talk about. But first, you were also interested in how, medications can actually affect these channels.
Right, because if we can control these channels, then we might be able to treat a bunch of conditions, right?
Yeah. Exactly. And that's what researchers are trying to do. You've probably heard of calcium channel blockers.
For high blood pressure, really.
Yep. They stop calcium from getting into those smooth muscle cells in blood vessels.
So the blood vessels can relax.
Yeah. And that lowers the blood pressure. And that's just one example. There's also research being done on targeting calcium channels to treat epilepsy, chronic pain, even some neurological disorders. But before we go too far into all that, maybe we should step back and look at the calcium signaling pathway. What happens once the calcium enters the cell.
Right. Because I'm sure it doesn't just float around in there. Right.
Definitely not. But we can talk more about that after the break. So yeah, we were talking about what happens once the calcium gets into the cell.
Because I'm assuming it doesn't just hang out in there doing nothing.
Right. It's not just floating around. It's a very organized process. It's like those calcium ions are looking for specific targets, like a lock and key.
So they bind to certain proteins.
Exactly.
Yeah.
And one of their main targets is this protein called calmodulin.
Calmodulin.
Yeah, it's when calcium binds to it, it changes its shape. And that allows it to interact with other proteins, a whole bunch of them, actually.
So it's like a domino effect.
Yeah, kind of. And one of the big ones that gets activated by this, calcium calmodulin complex is called KMK.
KMK.
Yeah. It stands for calmodulin-dependent protein kinase. Kind of a mouthful, but it's basically a molecular switch.
A molecular switch. What do you mean by that?
Well, when it gets activated, it can attach a phosphate group to other proteins. It's called phosphorylation.
Phosphorylation, right.
And that little phosphate tag can either, turn a protein on or off. It changes how it acts.
Oh, that's interesting. So it's kind of controlling all sorts of processes in the cell.
Exactly. And remember how you were asking about synaptic plasticity? Well, KMK plays a role there, too. It can, modify the strength of connections between neurons, which is how we learn and remember things.
Wow. So these calcium channels are involved in the long game too.
Yeah.
It's not just the immediate response.
Exactly. They're involved in those long-term changes as well. Now, you mentioned a paper that was specifically talking about muscle contraction. Do you remember those L-type channels we talked about earlier?
This is in the muscle cells, right?
Yep. So when calcium goes through those L-type channels, it starts a whole process that involves these two proteins, actin and myosin.
Okay, I remember those names from biology class.
Well, they basically interact with each other to form what are called cross bridges.
Cross bridges.
Yeah, and that's actually what makes the muscle fibers shorten so they can, generate force.
So it's like this tiny tug of war happening inside the muscle cells.
Yeah, good analogy. And all thanks to calcium doing its thing. And it's not just muscle contraction. Calcium signaling is also super important for releasing hormones from glands, controlling gene expression, even programmed cell death.
Wow, it's involved in a lot.
It's like the ultimate multitasker. And what's cool is that, the basics of how it all works are kind of the same across different cells, but the details can vary.
So different cells have their own variations on the calcium theme?
Right. So that means they can have all sorts of different responses, depending on what they need to do.
That's fascinating.
Yeah.
But I have another question. You've been talking about calcium coming into the cell, but what happens to it once it's, done its job?
Great question. Because it can't just stay in there forever, right?
Right. The cell would be overloaded with calcium.
Exactly. So there's a system for, getting rid of it, or at least storing it.
So there's a cleanup crew for calcium.
Exactly. There's special pumps and exchangers that move those calcium ions out of the cytoplasm.
Cytoplasm.
Oh, yeah. That's like the jelly stuff inside the cell around all the little organelles. Anyway, these pumps can either send it back outside of the cell, across the membrane, or into these little storage areas like the endoplasmic reticulum.
So the whole cycle comes in, does its job, gets sent out or stored away.
That's the idea. And keeping that balance is super important, You don't want too much calcium building up.
Makes sense. This whole system is so complex. It's crazy to think about it all happening inside every cell in our bodies all the time.
It is pretty amazing, right?
It is. But there's one more thing I'm really curious about, that whole idea of synaptic plasticity and how it relates to learning and memory. It's just mind-blowing to me that our brains are constantly rewiring themselves based on what we learn.
Yeah, it really is. And those calcium channels are a big part of that whole process. They're not just controlling the flow of calcium, they're shaping how we learn and remember and, interact with the world around us.
So we're back and ready to dive into, synaptic plasticity. It's just so cool how our brains are constantly rewiring themselves.
It really is remarkable, and those calcium channels are, right in the middle of it all.
Okay, so before we go any further, can you just remind me what synaptic plasticity is exactly? And how do those channels fit in?
Sure. So synaptic plasticity is it's basically how the connections between neurons, those synapses, can get stronger or weaker over time.
Right, like our brains are always adapting.
Exactly. And remember how we talked about, calcium influx triggering neurotransmitter release?
Uh-huh.
Well, that's just the first step, because calcium also plays a big role in those long-term changes that happen in synaptic plasticity.
So it's not just the immediate response, it's also involved in making those connections stronger or weaker over time.
You got it. So when a synapse is activated over and over, it can trigger this process called long-term potentiation, or LTP.
LTP.
And that basically strengthens the connection, making it more likely that, the signal will get through. And guess what? Calcium is a key part of making that happen.
So it's like calcium saying, hey, this connection's important, make it stronger.
Exactly. It sets off all these signaling pathways that lead to, actual physical changes in the synapse.
So it becomes more efficient at transmitting those signals.
Yep. And it's pretty amazing how something as small as calcium can have such a big impact on how we learn and remember things.
It really is.
Yeah.
And you mentioned that calcium can also be involved in weakening those connections, right? Like with long-term depression.
That's right. LTD. So it's like a two-way street. Calcium can either strengthen or weaken connections depending on what's going on.
So it's all about balance.
Exactly. It's all about, making sure the brain is working efficiently.
Now, you keep mentioning these signaling pathways. Can you give me a specific example of what one of those looks like?
Sure. So one of the most important ones is the CAM-KII pathway.
CAM-K, yeah.
Yeah. It stands for calcium calmodulin-dependent protein kinase 2. It's an enzyme that plays a huge role in both LTP and LTD.
Okay, so how does it work?
Well, remember calmodulin?
The protein that binds to calcium.
Yep. So when there's a lot of calcium in the neuron, it activates CAM and KII, and then CAM-KI goes and attaches this phosphate group to other proteins.
Right, phosphorylation.
And that can, change what those proteins do, where they go, even how much of them there is. It's pretty powerful stuff.
So CAM-KI is basically in charge of all these changes happening in the synapse based on those calcium signals.
Exactly. And one of its main targets is these things called AMPA receptors.
AMPA receptors.
Yeah. They're important for transmitting their signals between neurons. So if you change how many there are or how they work, it can really affect how strong the connection is.
Right, because if they're involved in transmitting the signal, then changing them would definitely have an effect.
Right. So in LTP, CAM-KI can actually make the synapse have more AMPA receptors.
So it's like, hey, we need more of these to make this connection stronger.
Exactly. But in LTD, it can do the opposite.
Take away some of the AMPA receptors.
Yep. So it weakens the connection.
It's incredible how much control this one enzyme has.
It really is. And that's just one pathway. There are tons of others that are involved in synaptic plasticity, each with its own, little role to play.
So it's this whole web of interactions constantly shaping and reshaping our brains.
That's a great way to put it. And we're still figuring out all the details, but that's what makes this research so exciting.
I totally agree. This whole deep dive has been fascinating. We started with those tiny channels,
Yeah.
followed the calcium, and now we're talking about how it all affects learning and memory.
Yeah. I mean, it really shows you how these microscopic processes can have such huge effects on, everything.
It really does. Well, thanks for joining me on this deep dive into the world of voltage-gated calcium channels. I think we all learned a lot today.